Good morning, friends. First of all, I welcome in the Sanjudi's Marvelous Apologies online video YouTube class. Today we are going to see the BSc first year class paper number 3, the unit Microbial staining techniques. It is very important staining techniques because microorganisms, they are very small sizes, they cannot observe with our naked eyes. They can always observed under the microscopes. They are basically colorless, and for that purpose it is very important to colorize this microorganism by using different stains under the microbial staining techniques. So in this unit we'll see the first definitions of different parts of the microbial staining techniques. First definitions, we'll see the staining. It is the process of giving color to particular microorganism, and its component is known as staining. Because we can stain the different microorganisms for observations, we can stain. Due to the staining process, what happens, we can observe the specific shape of microorganisms. We can observe specific part of the microorganisms in the staining techniques. So what is meant by stain? Stain, it is an organic biological compound containing benzene ring and also chromophore and exochrome group. Stains are used commonly in microbiology to increase the contrast between microorganism and its part of its background, due to which what happens, they can easily visible under the microscopes. So what is meant by dye? Dye, it is a chemical colorizing agent, or it is a general colorizing agent. They cannot use in the biological purposes. So after that, in the dye or in the stain, there are three basically components are present. One is the benzene ring, then second one is the chromophore group, and third one is the exochrome. Let's see one by one. The first, the benzene ring. Benzene ring is the colorless part of the dye, and it is a basic structural component of dye. So what is meant by chromophore group? Chromophore group, it is the functional group of dye that gives color to the stain. It is very important because whatever the parts of microorganisms, they are colored due to the chromosome group, because it fixes the color to the microorganisms. Exochrome, it is the group that gives ionic property to the stain, that is called as exochromes. After that, we'll see the definition of acidic stain, basic stain and neutral stain one by one. What is meant by acidic stain? Acidic stain is also called as anionic stains because acidic stain is the stain which contain negatively charged chromogens and which can generally stain the positively charged components such as the background. Why? Because microorganisms, the cell wall of the microorganism or the different parts of the microorganism, they are basically negatively charged, and acidic stain is also negative charge. Negative charge, there is the repulsions. They cannot colorize. That's why acidic stain generally color the background. For example, nigrosin, Indian ink and eosin. Basic stain generally contain the positively charged chromosomes. That's why they can color the microorganism, its parts, because they have the negative charge. That's why there is the attraction between these two charges. That's why they colored the bacterial cells. For example, methylene blue, safranin, malachite green, basic fuchsine crystal violet. These are the different example of basic stain. Neutral stain. In a neutral stain, both cations and anions are colored such as that net charge is neutrals. They are equal, that's why they are the neutral. For example, these are strains. After that, we'll see the modern stain. It is a chemical that generally forms an insoluble complex with the stain and fixes it or causes the stain to penetrate more deeply into the cell. These are the used in indirect staining techniques. So after that, we'll see the definition of chromogens. What is meant by chromogen? Chromogen, it is a substance which can be readily converted into dye or other colored compound, because whatever the stain they are colored, it is due to the chromogens. After that, we'll see the definition of leuco compound. It is a colorless organic compound formed by the reduction of dye and giving dye or color again by oxidation process. They are called as leuco compound. Next definitions, then fluorochrome. It is also a substance that can excited by certain light sources. Means just like stars observes that type of the microorganism or its part, they are shining under the microscope due to the fluorochrome stains. Next, the decolorizing agent. What is meant by decolorizing agent? It is a chemical used to remove the excess stain in indirect staining method. For example, ethanol. They are generally used to remove what? The excess stain. And last definition of these stains or staining techniques is the counter stain. Counter stain is a very important stain because it is stains with color contrasting to the principal stain, making the stain structurally easily visible using a microscope. Counter stain why used? Because for clearly observations of microorganism under the microscope, the counter stains they are generally used. Due to the use of this counter stain, what happens? We can clearly and we can shiningly observe these microorganism under the microscope. So friends, these are the different definitions of microbial staining techniques. In the next lecture, we will see the different methods or different techniques of the microbial staining techniques. Till then, bye.